Welcome to this explainer. Now, whether you're gearing up for a complex university research project or diving straight into advanced analytics, there's an absolute truth we need to establish right out of the gate. A biology lab, it's a completely different environment, and the stakes are high. Treating it with the utmost respect is literally your very first step to doing some truly fascinating science.
Okay, let's dive into this. Ask yourself, are you truly ready for the lab? Because once you step inside those doors, you aren't just dealing with textbooks anymore. You're working alongside potent chemicals, razor-sharp instruments, and unpredictable biological hazards that absolutely demand your full attention. The rules we're going to discuss today, they are completely non-negotiable. They're the essential requirements for you to actually survive and thrive in this totally unique environment.
So here's the chronological flow of our lab journey today. We'll kick things off with a quick welcome to the lab, armor up with essential PPE, navigate handling chemicals and biohazards, and then cover fire and physical safety. After that, we'll hit general rules and cleanup, and finally, wrap up with handling lab emergencies.
Section 1. Welcome to the Biology Lab, Serious Responsibilities for Fascinating Science.
All right, Section 2. Armor up, essential PPE. This is your first line of defense.
Before you even touch a beaker, there are 3 absolute essentials for your protective gear. First up, you need a fully buttoned lab coat. And I mean fully buttoned, not left open casually like a jacket. You need maximum coverage. Second, you must wear chemical splash safety goggles. Guys, regular prescription eyeglasses absolutely do not count as eye protection here. They don't seal around your face so they won't stop a splash. You need those wrap-around chemical splash goggles. And third, you've got to wear nitrile or latex gloves. Always remember to check them for little holes before you begin.
Now, what's really interesting about this slide is you probably don't think twice about what your lab coat is actually made of, right? But it's literally a matter of life and limb. The stark difference here is between safe fabrics, like cotton or fire-resistant materials, and incredibly dangerous synthetics. The terrifying reality of fabrics like synthetic nylon or polyester is that if there's a fire, these materials will literally melt directly onto your skin. It causes catastrophic burns. So always make sure your coat is cotton or flame-resistant. No exceptions.
Okay, so your experiment is done. But taking your gloves off improperly, that can completely ruin all the protection they just provided. Let's walk through this critical glove peel technique. It's pretty simple. You pinch the outside of the first glove near the wrist, peel it downwards so it turns inside out, which effectively traps all the nasty chemicals inside. Then, slide a clean, ungloved finger under the wrist of the second glove, peel it off, and throw them straight in the bin immediately. Doing it this way guarantees you never spread chemical or biological contamination to your bare hands.
Moving on to Section 3. Handling chemicals and biohazards, navigating dangerous science.
Now that we're properly geared up, we enter the active experiment phase. You know, in a lab, a highly corrosive acid or really toxic solvent can look exactly like a glass of plain tap water. Clear liquids are incredibly deceptive. That makes it absolutely vital to read every single label before you even pick up a bottle. And you've got to understand these specific hazard classifications. A flame symbol, that means flammable. A test tube spilling on a hand means corrosive. A skull indicates toxic, and an exclamation mark means irritant. Never, ever assume what a substance is based purely on its appearance.
Here's a quick, highly practical pro tip for you. Waft, don't sniff. If you absolutely need to detect the odor of a chemical, never hold the bottle directly up to your nose. You could inhale a massive lungful of toxic fumes, and trust me, you don't want that. Instead, hold the container away from your face and gently use your hand to wave or waft a small amount of the vapor towards your nose.
So, the crucial point here is, never pipette by mouth. It sounds like a joke, doesn't it? But historically, people actually used to do this. Imagine treating a glass tube full of some toxic carcinogenic lab liquid like a straw for your favorite iced coffee, just sucking the liquid up with your mouth. It is shockingly dangerous. It's completely forbidden in any modern lab, and obviously it can result in severe poisoning. Always use a rubber bulb or mechanical pump, period. When we move from chemicals over to biology, a golden rule applies. You must treat all biological materials as potentially hazardous. Honestly, it doesn't matter if it's just a simple environmental soil sample, some mold growing on a plate, or animal blood. You have to strictly assume that it contains unknown pathogens. Wear your gloves, never touch your face, and always wash your hands after handling any living organism or tissue sample.
Section 4. Fire and physical safety. Controlling the physical environment.
1500 degrees Celsius. That's the temperature a Bunsen burner flame can reach. Pretty intense, right?
This brilliantly illustrates exactly why tying back long hair and rolling up loose sleeves is an absolute, non-negotiable mandate. Loose hair catching fire is literally one of the most common and most terrifying lab accidents out there. You have to control your physical profile before you even think about striking that match. And when it comes to handling sharp instruments like scalpels and glass, the rules are incredibly rigid. Always cut away from your body. Keep those scalpel blades capped when you aren't actively cutting. Never, ever pass a scalpel to a lab partner blade first. Just set it down on the bench and let them pick it up themselves. And hey, if a beaker shatters, never use your hands to clean it. Grab a brush and dustpan. Remember, in a biology lab surrounded by microorganisms, even a tiny, seemingly harmless paper cut can become dangerously infected.
All right, Section 5, General Rules and Cleanup, wrapping up the lab session.
I really want to contrast two incredibly strict bans in the lab. No food or drink, and no cell phones on the bench. Understanding the why here is totally essential. We ban food because lab air and benches have toxins. If you eat, you're ingesting those toxins, plus your crumbs are going to ruin sensitive experiments. But what about cell phones? Well, cell phones are major contamination vectors. Think about it. If you touch a contaminated bench, then touch your phone, and then later hold that same phone right up to your face, you have completely bypassed every single piece of protective gear you wore all day. Keep your phones packed away in your bags.
So, once your workstation is fully wiped down, tools are put away, and everything is pristine, you arrive at the absolute final, mandatory step before walking out that door. Hand washing. Wet your hands with clean, running water. Apply soap and lather well, then scrub for at least 20 continuous seconds. Don't rush this part. Finally, rinse thoroughly and dry. Even if you wore your gloves perfectly, this is your final failsafe to ensure you leave all the toxins inside the lab exactly where they belong.
Section 6. Handling lab emergencies. Preparedness is survival. Let's move to this next part and see how it builds, because the golden rule of emergencies is that you must know exactly what to do before an incident occurs, not during the chaos and panic of an active emergency. You really need to develop a habit here. Perform a 10-second mental safety scan at the very start of every single lab session. Stand at your bench, look around, and physically locate the fire extinguisher, the fire blanket, the eye wash station, the emergency safety shower, the first aid kit, and your emergency exits. It only takes 10 seconds, and it could literally save your life or the life of a colleague. To really put into perspective why this matters, let's look at this high-stakes timeline of a chemical eye splash. At 0 seconds, the chemical hits. Within 2 seconds, you must be shouting to alert your teacher or peers. Within 5 seconds, you need to reach that eye wash station you already located in your safety scan. And then, you have to flush your eyes continuously for 15 agonizing minutes. This timeline perfectly demonstrates why you can never, ever remove your safety goggles in the lab, not even for a second. And that brings us to our final thought for today's explainer. Welcome to the lab. Stay safe and enjoy the science. Lab safety rules aren't here to restrict you. They exist to empower you. By respecting your environment and protecting yourself, you unlock the freedom to safely discover the absolute wonders of the biological world. But I want to leave you with this question to think about. The next time you step up to a lab bench, will your habits be the thing that saves a critical experiment or the thing that causes a complete evacuation? Stay safe and enjoy the science.